Assalamu alaikum wa rahmatullah. My name is Ziya Al-Hamdan, and today we're going to be discussing the parathyroid physiology. Looking at the contents of the presentation, we have two major components we'll be covering today: the parathyroid hormone and vitamin D. And under each of these, we have some specific objectives or learning goals, and we'll be covering those too, inshallah. So, as an introduction, you guys should know that we have 4 parathyroid glands, and they are largely composed of chief cells. And if we have a look at the figure, you can see the 4 glands, and they're all sitting on the posterior side of the thyroid gland. And they're either sitting on the upper pole of the gland or on the lower pole. So, if we look closely into one of the glands, you can see the chief cells, those purple cells, and they make up most of the gland. One might ask or might have noticed that we have another type of cell, which is called an auxoval cell, and the function of an auxoval cell is not yet certain, but they are believed to be modified or depleted chief cells that no longer secrete the hormone. What hormone are we talking about? We're talking about the parathyroid hormone, and the chief cells are the one responsible for the synthesis and the secretion of the hormone. So what does regulate this hormone? We have the calcium, we have vitamin D, and we have phosphorus. All of these regulate the secretion of the parathyroid hormone. Okay, let's move into the parathyroid hormone. Okay, so we'll start with the synthesis of the parathyroid hormone. So the synthesis begins with the production of the pro-parathyroid hormone in the rough endoplasmic reticulum. And you can see that this pre-pro protein has a sequence which is called the signal sequence, or the pre-fragment. This fragment targets the parathyroid hormone to be transported into the lumen of the endoplasmic reticulum. There, in the lumen, this signal sequence is cleaved, resulting or producing the pro-parathyroid hormone. And then this pro-parathyroid hormone is transported into the Golgi complex or the Golgi apparatus. There, the pro sequence is then cleaved, and this produces the mature or the intact parathyroid hormone. And this intact parathyroid hormone is then stored into vesicles or granules, secretory granules. Okay, in the secretory granules, the cleavage of the pH or the PTH begins, and the cleavage yields 2 fragments, an N-terminal and a C-terminal. Okay, so the half-life of the hormone is about 4 minutes, but then the question comes, how does it produce its effect on the biological system? And if we want to know the answer, we need to have a look at the figure which I've shown you before, because the answer lies there in the N and the C terminals of the parathyroid hormone, as you can see here, this N terminal and this C terminal. Okay, so let's look at the properties of each of these terminal peptides. As for the N-terminal, it is 33 or 36 amino acids, but the C-terminal is much longer than that. And for the N-terminal, almost all of the known activity of the parathyroid hormone resides in this N-terminal. On the other hand, the C-terminal is not active. The N-terminal is or has a short half-life and is hydrolyzed in the kidneys. And as for the C-terminal, it is of longer half-life, has longer half-life of that of this N-terminal, or even the active, intact parathyroid. Okay, so the major stimulus for the secretion of the parathyroid hormone is a decrease in the blood and extracellular fluid concentration of calcium, or hypocalcemia, you can say. So apparently, this hypocalcemia does not only stimulate parathyroid secretion, but also stimulates the synthesis of new parathyroid hormone. But is this necessary? Well, the answer is yes, it is necessary, because the parathyroid gland contains only enough parathyroid hormone to maintain stimulated secretory response for only several hours. So we always need to make more and more of the hormone. So what's the mechanism behind this? Okay, the mechanism lies in this receptor, which is called the calcium sensing receptor, CaSR for short. Okay, this receptor is a member of the G protein coupled receptors and is found mainly in the chief cells again. Okay, so the receptor is coupled to G alpha Q, which has diacylglycerol and inositol triphosphate and calcium as second messengers. So calcium binds to the receptor and activates it. This ultimately results in the inhibition of parathyroid secretion. In this figure, you can see that when calcium binds to the receptor, it activates a sequence of events, and ultimately resulting in the production of the inositol triphosphate, the diacylglycerol, and calcium, and all of these are the second messengers. This ultimately leads to the inhibition of the release of the parathyroid hormone, and of course the synthesis too. So, the parathyroid hormone receptors. The parathyroid hormone has two receptors. One is called parathyroid hormone 1R, and the second is 2R. Okay, simple. So, the regulation activity of the parathyroid hormone, which is the regulation of the calcium, is mainly through the first receptor, or 1R. However, this 2R role in regulating calcium is still unknown. Okay, so 1R receptor has the highest abundance in kidneys and bones. So in kidneys, it is abundant in the proximal and distal convoluted tubules, and in the bones, mainly the osteoblasts. So let's discuss this receptor a bit more. It is a G protein coupled receptor that is coupled to two G proteins, G alpha Q and G alpha S, meaning that it has two signal transduction pathways. It binds the N-terminal fragments, of course, the active part of the parathyroid hormone, and a peptide which is called the parathyroid related peptide. So then what happens when those receptors are activated? You can divide the effect on three parts: the effect on bones, the effect on kidneys, and the indirect effect on the intestines. This ultimately increases the serum calcium, and decreases that of phosphate. Okay, so let's discuss each organ separately, starting with the bones. Okay, the parathyroid hormone promotes both bone resorption and bone synthesis. This needs to be explained. So, the persistent increases or the chronic increases of the parathyroid hormone on bone results in bone resorption. However, the intermittent increases in plasma parathyroid hormone have predominantly bone synthetic effects. Okay, so the surface receptors of the parathyroid hormone are expressed on osteoblasts and not osteoclasts. Keep that in mind. only on osteoblasts. So it increases the serum calcium concentration in two phases. One is called the rapid phase and the other is called the slow phase. During the rapid phase, mobilization of the calcium from the internal bone stores happen. And during the slow phase, we have the liberation of both calcium and phosphate. And this happens through bone resorption. Okay, so there's this system which you should guys know, which is called the Osteocytic Membrane System. Okay, before discussing this system, when we think of the osteoblasts or the osteocytes, we usually think or tend to think of their osteoblastic nature or the bone forming nature. However, it has been shown that they also, the osteocytes and the osteoblasts, they contribute through this system, the osteocytic membrane system, to bone salt absorption. Wow. So let's get to know the system. Okay, so this system is formed by both the osteoblasts and the osteocytes, and it provides a membrane that separates the bone from the extracellular matrix. Okay, in this slide you guys can see the osteocytic membrane system, and those are the osteoblasts on the outer surface, and again some other osteoblasts on the inner surface close to the blood vessel, and between those osteoblasts we have the osteocytes sitting in the center, 4 of them actually. Okay, so the question comes, where does parathyroid hormone fit here? Okay, so apparently the parathyroid hormone activates the calcium pump found in the cells of the osteocytic membrane by increasing the permeability of its bone fluid side. Subsequently, calcium diffuses from the fluid to the membrane cells, leading to the stimulation of the calcium pumps on the other side and the expulsion of calcium ions. As we can see in the figure, the parathyroid hormone acts somewhere here and here on the side of the cells which is facing the bone fluid. This leads to the entry of calcium ions into the cells. When calcium enters the cells, the concentration of calcium increases. This leads to the activation of the calcium pumps on the other side, leading to the expulsion of the calcium ions outside of the cell. Okay, so we have the slow phase. Okay, in this phase, the parathyroid hormone acts on the osteoblasts and osteocytes. This releases certain factors. Okay, subsequently, these factors indirectly activate osteoclasts. So it does not directly activate osteoclasts, but through osteocytes and osteoblasts. Okay. The osteoclasts activation occurs in 2 stages. We have an immediate activation of the already existing osteoclasts and stimulation of formulation of new osteoclasts. So, what about bone formation? So, like we said, the intermittent secretion of parathyroid hormone leads to or favors bone formation. Okay, so parathyroid hormone can promote bone synthesis by 2 mechanisms. We have a direct mechanism and an indirect mechanism. In the direct mechanism, the hormone activates calcium channels in the osteocytes, or another name, in osteocytic osteolysis. The calcium transfer from bone fluid and then to the osteocyte, and then through gap junctions to the osteoblasts, and then to the extracellular space. This contributes to mineralization. And indirectly, in the osteocytic bone resorption, or the osteoclastic bone resorption, when osteoclasts start resorbing bone, this leads to the release of growth factors residing within the bone itself. Growth factors like insulin like growth factor 1 and insulin like growth factor 2, and the transforming growth factor beta. All of these are residing within the bone itself, and they're being released following bone resorption. Okay, now we'll be discussing the effects of the parathyroid hormone on the kidneys. And let's review, first of all, let's review the nephron. Okay, here in the nephron you can see the glomerulus and the Bowman's capsule, and then from the Bowman's capsule we move into the proximal convoluted tubule. Okay, and then the loop starts. The loop of Henle, we have a descending and the descending limb and the ascending thick limb. Okay, keep that in mind. And we have the distal tubule, and then from the distal tubule the filter is collected in the collecting ducts. So parathyroid hormone promotes the reabsorption of calcium ions, it inhibits the reabsorption of phosphate, and it stimulates the last step of the synthesis of the vitamin D, or the active vitamin D. And we'll be talking about each of these separately. Okay, let's start with the calcium reabsorption promotion. Okay, you guys should know that most of the filtered calcium, about 65%, is passively reabsorbed in the PCT, or the proximal convoluted tubule, and about 25% in the thick ascending limb of Henle, the loop of Henle. So the parathyroid hormone acts on the cortical thick ascending lip of loop of Henle and the distal convoluted tubule. So again, the reabsorption passively happens in the PCT and the thick ascending limb, and mostly in the PCT. However, parathyroid hormone acts on the thick ascending limb of loop of Henle and the distal convoluted tubule. Okay, the phosphate reabsorption inhibition. The parathyroid hormone inhibits phosphate reabsorption in two places. We have the proximal convoluted tubule, or the PCT, and the distal convoluted tubule, or the DCT. So the parathyroid hormone decreases the activity and internalize and degrade the sodium phosphorus contrasporter in the luminal membrane of the proximal tubules. Okay, looking back at the nephron, we can summarize what we've discussed about the effects of the parathyroid hormone in the kidneys. Okay, so the phosphate reabsorption is inhibited in two places, the distal convoluted tubule and in the proximal convoluted tubule. However, on the other hand, we have the reabsorption of calcium. The reabsorption is promoted in two places too, in the thick ascending limb of the loop of Henle and in the distal convoluted tubule. So keep that in mind. Let's move into the indirect action of parathyroid hormone on the intestines. So how does PTH help in the synthesis of calcitriol, or the active form of vitamin D? Mainly through two mechanisms. We have an increase in the synthesis of 1-alpha hydroxylase, okay, in the proximal tubules. This enzyme converts calcidiol into calcitriol, or the inactive form of vitamin D, into an active form. Okay, and PTH also decreases the activity of 24-hydroxylase, which inactivates calcitriol, or the active form of vitamin D. And you can imagine how important the second step is in the states of vitamin D deficiency. So this ultimately results in an increase in the active form of vitamin D. This active form enhances both calcium and phosphate absorption in the intestines. Okay, in this slide we'll be summarizing the effects of PTH. Like we said, a decrease in the calcium concentration, or hypocalcemia, lead to the activation of the calcium sensing receptor in the chief cells, which are located in the parathyroid glands, the 4 glands here. Okay, this leads to an increase in, or the release of, parathyroid hormone in the blood. It has two main actions, on the bone and on the kidney. In the bone, we have an increase in bone resorption and calcium efflux out of the bone, and in the kidney we have the promotion of calcium reabsorption and the inhibition of phosphate reabsorption, and also through the activation of 1-alpha hydroxylase and the inhibition of 24-hydroxylase, we ultimately result in an increase in vitamin D, or the active form of vitamin D, in the blood. This in itself stimulates the kidney to absorb calcium and phosphate, both. So all of these three actions lead to an increase in calcium. Okay, let's move into vitamin D. Okay, so vitamin D itself is not active, and it must be converted to the active form in order to exert its effects. Okay, and the vitamin itself is fat soluble and exists in two forms. Actually, vitamin D is one of the 4 soluble or fat soluble vitamins. We have A, we have K, we have E, and we have D. You can memorize it as in أكيد بالعربي. Okay, so let's go back to the two forms. We have vitamin D3 or cholecalciferol. It is mainly formed from 7-dehydrocholesterol, but can also be obtained from different sources. And for vitamin D2 or ergosterol, this can only be obtained from dietary sources and mainly vegetables. So vitamin D2 and vitamin D3 differ only in the side chains of ring D, as in vitamin D. Okay, so the side chain in vitamin D3 or cholecalciferol is characteristic of cholesterol, whereas that of vitamin D, ergocalciferol, is characteristic of plant sterols. In this slide, we can see the different forms of vitamin D3 in our body. Okay, so it starts off with a compound named 7,8-dehydrocholesterol, and then through the action of the sun, we get pre-vitamin D3. This pre-vitamin D3 is converted into vitamin D3 or cholecalciferol. This compound then circulates the blood and it reaches the liver. In the liver, we have an enzyme which is called 25-hydroxylase. This enzyme converts cholecalciferol into calcidiol or 25-hydroxyvitamin D3, and then it goes to the kidneys. In the kidneys, we have this enzyme which we talked about previously called 1-alpha hydroxylase. This enzyme converts calcidiol into calcitriol, the active form of the vitamin. And then after exerting its actions, we have the enzyme 24-hydroxylase. This enzyme inactivates the active form of the vitamin or calcitriol and converts it into calcitriolic acid. Here is another figure which basically says the same thing: the action of the ultraviolet ray, exerting it on the 7-dehydrocholesterol, then pre-vitamin D3, and then it circulates the blood, reaches the liver, 25-hydroxylase, then to the kidneys. In the kidneys, 1-alpha hydroxylase. 1-alpha hydroxylase yields our active compound, and then we have the action of 24-hydroxylase and resulting in this compound and then excretion. Simple. So let's move to the effects of vitamin D. Vitamin D acts on several tissues, of which we have bones, we have kidneys, we have intestines. Again, let's take each one at a time, starting with bones. So vitamin D plays a role in both the resorption and the deposition. The resorption happens through the high concentration of the high amounts of vitamin D, and small amounts plays a role in the deposition of bone. Okay, so the mechanism behind this is unknown, although some believe it's because of increased calcium transport across the membrane. So note that in the absence of vitamin D, the action of parathyroid hormone on bones, causing their resorption, is greatly reduced. So we need the contribution of vitamin D. And some say it's even prevented. You can now imagine how important vitamin D is. Okay, the effect of the vitamin on the kidneys and the intestines. So vitamin D increases the reabsorption of both calcium and phosphate. However, this effect is weak and hardly plays a role in the regulation of extracellular fluid concentration. However, in the intestines, it increases the absorption of both phosphate and calcium, same as parathyroid. Okay, in this slide, we will be talking about vitamin D and its effects on the intestinal calcium absorption. So, in the intestine, calcium is absorbed by two mechanisms, a paracellular mechanism and a transcellular mechanism. The paracellular mechanism is the predominant one. However, it is not under the control of active vitamin D. This one happens all across the intestine. Let's move into the transcellular mechanism. The transcellular mechanism occurs only in the duodenum. However, it is under the control of active vitamin D. So what happens exactly? Calcium enters the cell through the apical membrane, okay? Then inside the cell, calcium is buffered with several proteins. One of these is calbindin. Some of the calcium ions are being taken up by cellular organelles such as the endoplasmic reticulum. Okay, so after the calcium has been buffered, okay, it exits the cell by two other mechanisms, either by a calcium pump, which requires ATP, or by a calcium-sodium exchanger, and both of these occur at the basolateral membrane. So this form of transport, the transcellular transport, those three steps are all controlled by the active form of vitamin D. Let's move to the effects of vitamin D on the intestinal phosphate absorption. So apparently vitamin D increases the number of sodium phosphate cotransporters, which are located on the apical membrane. So as the name implies, those channels co-transport sodium and phosphate. So then phosphate exits the cell across the basolateral membrane. The net effect is phosphate absorption. And yes, finally, those are my references. You can check them out, guys. Yes, thank you for your patient listening. I hope you enjoyed the video.